The Autism Research Institute relies on the generosity of donors like you to make this webcast possible. If you enjoy this presentation, please consider making a donation. Thank you.
Okay, well, thank you very much, Denise, and it's a great pleasure to be here today. And I'm going to be talking about inflammatory mechanisms and experimental therapeutics of autism spectrum disorders. I'll give a little bit of an overview also in terms of inflammatory mechanisms as it relates to some other neuropsychiatric disorders. And I'd like to acknowledge funding here and disclosure. We did receive some funding from the Simons Foundation for our study of TSO in autism and for our study of hyperthermia in autism. And we received drug and matching placebo from Coronado Bioscience for the TSO study as well. First, I would say that in many of the neuropsychiatric and developmental disorders that we deal with these days, we're starting to think that many of these conditions are really gene by environment interactions. And I think that gene by environment interaction may play an important role in the rising prevalence that we see with the most recent CDC estimates of ASD are 1 in 68. Also, we think that cytokines play an important role in many of these neuropsychiatric and developmental disorders, and that one sort of endpoint of cytokine-induced inflammation is activation of microglia in the brain. I'm going to talk very briefly about PANDAS, these pediatric autoimmune neuropsychiatric disorders associated with strep, as a model of repetitive behaviors, and then we'll focus on autism spectrum disorders as well. First, I think that it's important to recognize that in many of the autoimmune disorders that we see in medicine, patients can often have a new onset or an exacerbation of other CNS or neuropsychiatric features. So a classic example of that, for example, is lupus. And so, you know, we're starting to see a lot more patients with autoimmune illness who are presenting with neuropsychiatric kind of symptoms. Also, some of the common treatments that we use in psychiatry, like antidepressants, actually have a mechanism of action where they can modify inflammation. And it's well known that when pro-inflammatory stimuli, like interferon, for example, is administered to treat hepatitis, this can induce psychiatric symptoms like depression. Now, some of the disorders that we had looked at in the past from, let's say, a psychoanalytic perspective, our recent understanding has really radically changed. So, for example, gastric ulcers used to be felt that these were a manifestation of underlying psychodynamic conflict. And more recently, a particular infectious agent, H. pylori, has been identified, and that in many patients with ulcers, you can treat this with antibiotics. That's an example of a change in the way that we think about this sort of brain-immune connection. One important concept and something that's been the focus of research interest recently is our understanding of the microbiome. The idea that we have more cells in our body that are actually not us than are us. And in fact, by far the vast majority of DNA in our bodies are not human DNA, it's microbial DNA. So you can think of us as, in fact, being these superorganisms that are made up of thousands of different species, including humans, but where there's other parasites and bacteria and viruses that also found within the human body and make up much of the DNA that's present in the human body. And in particular, people have focused on the microbiome and the important role that that plays in a number of chronic disorders that we deal with these days, including obesity and type 1 and type 2 diabetes. Also in medical practice these days, there are a number of common laboratory blood measures that give us sort of a biomarker, or they give us an understanding of inflammatory mechanisms that could be occurring in the body. And much of the focus these days is the impact of chronic inflammation in mediating a range of medical illness. So, for example, in routine blood work, we can measure inflammatory cytokines, things like TNF-alpha or IL-6. We can measure acute phase proteins like C-reactive protein. And the recent study in disorders such as depression or schizophrenia suggests that those individuals who have these biomarkers of inflammation, high levels of C-reactive protein greater than 5, may actually have sort of a unique response to anti-inflammatory treatments. We can measure things like the ESR, the erythrocyte sedimentation rate. We could look at the blood count and the white blood cell and eosinophil counts. We can measure anti-strep antibodies in the blood, like ASO antibody and DNase B antibody. And nowadays there are new measures also where we can look at antibodies that cross-react with brain tissue, or anti-brain antibodies. And these can all be measured now in routine medical care. Cytokines are things that are in the bloodstream and in the spinal fluid or in the central nervous system tissue that may be mediators of inflammation. And pro- and anti-inflammatory cytokines affect multiple different pathways that are important for development or neuropsychiatric illness. So they play a role in neurotransmitter or monoamine metabolism. They influence neuroendocrine function. Cytokines play an important role in shaping synaptic plasticity, how different neurons communicate with each other, and the structure of synapses, for example. Cytokines play an important role in shaping brain circuitry, and chemokines play a role in attracting inflammatory cells from the periphery into the central nervous system. So, for example, if you look at the left, these anti-inflammatory cytokines like IL-4 play an important role in resilience and health. And they can modify T-cell function and macrophage function. They can affect the blood-brain barrier, and they can be associated with a brain-derived neurotropic factor or neuroplasticity. Pro-inflammatory cytokines, for example, like TNF-alpha, can be released from macrophages, for example, and it can cross the blood-brain barrier and be associated with activation of microglia in the brain. And this can also be associated with then a release of agents like MCP, which are associated with the ability of things like monocytes, for example, to be able to penetrate through the blood-brain barrier and then play a role in inflammation in the central nervous system, so that they can also be involved both in the periphery and in the CNS with the development of neuropathology. These cytokines, for example, in the periphery, in macrophages, for example, can influence tryptophan being metabolized to kynurenine, and kynurenine can cross the blood-brain barrier and act on microglia, where you can activate the glutamate system or activate NMDA receptors, which can be associated with things like neurotoxicity. And then a range of things may, such as stress, for example, or inflammation, can influence enzymes in the brain like tetrahydrobiopterin, or BH4, that are intimately involved in the synthesis of all of the central neurotransmitters. And this enzyme can go into an inactive state, and that can influence neurotransmitter synthesis. That's why administration of things like methylfolate, for example, can be helpful in sort of activating these enzymes like BH4 and regulating neurotransmitter synthesis. And then nowadays, we know that there are a range of factors. So abnormal immune function, or T-cell function, gut impairment, for example, or inflammatory bowel disease, stress, trauma, obesity, for example, that can be associated with chronic inflammation. And we know that many of the medical problems these days that doctors deal with are really the result of chronic inflammation. So diabetes, cancer, depression, cardiovascular risk may all be associated with chronic inflammation in humans. Recently, there have been some interesting epidemiology studies that have suggested that these gene-environment interactions may be very important, not only for developmental disorders, but for other disorders like depression. So, for example, in populations, the greater the number of significant infections that result in hospital care may interact with the presence or absence of a range of autoimmune illness, like lupus, for example, to be associated with significant increased odds ratio or increased risk for the development of things like major depression. So if you see, for example, that if you have individuals who have an autoimmune disorder and then have had several different kinds of infections, that there's an interesting gene by environment interaction or infection by autoimmune interaction, such as the odds ratio or the risk of developing a major depressive disorder is significantly multiplied. And so often we think about these sort of gene-environment interactions, and there can be environmental triggers, for example, like infection, that can kick off inflammatory processes in individuals that may be genetically vulnerable for the risk of things like autoimmune illness. And then this interaction can result in central nervous system or neuropsychiatric symptoms. So one idea is that in the old days, individuals used to die as a result of acute infections. People would die when they got an infected jaw from a toothache or something like that. And nowadays, people aren't dying in developed countries as much from acute infection, but we're suffering from the effects of chronic inflammation. So one idea is that the microbiome, this complex mix of different organisms within the body, and particularly in the gut, may play a role in regulating chronic inflammation, and that we can have sort of a healthy gut microbiome, or healthy gut parasites or bacteria, or we can have dangerous gut bacteria or parasites. But these healthy gut microbiota, or old friends, played a role really in dampening down chronic inflammation by regulating the immune system. And this actually has effects because chronic inflammation, may go through the vagus nerve and affect the central nervous system, for example, or chronic inflammation can be associated with transport of these pro-inflammatory cytokines across the blood-brain barrier, or chronic inflammation or cytokines can be associated with the ratio of T helper to T suppressors and influence trafficking of different components into the brain that can regulate CNS symptoms like depression, for example. So interestingly, sometimes acute inflammation could be associated with an improved mood, but the chronic inflammation often can be associated with things like depression or irritability or agitation or disruptive behaviors, for example. And that the idea is that these healthy gut bacteria or the microbiome may play a role with these sort of old friends that have evolved over many, many generations in regulating chronic inflammation in humans. Now, this may be more important these days as individuals have moved from rural regions into urban regions. And when people in developed countries or rich industrialized nations, for example, or urban areas, may have lost some of these favorable gut microbiota, and as a result of that, we see an increase in chronic inflammation and persistently raised pro-inflammatory cytokines. This can be associated, for example, with increased risks of obesity, for example, and can be associated with the development of certain autoimmune problems like inflammatory bowel disease or allergies, for example, diabetes, obesity, cardiovascular illness. And in the brain, increased trafficking of these pro-inflammatory cytokines can be associated with symptoms like depression or irritability. In the developing country or in rural areas where there's more of these healthy microbiome parasites, for example, there may be less chronic inflammation, and as a result of that, less of these pro-inflammatory cytokines and less activation of microglia, for example, in the central nervous system. So you may see different rates of illnesses like asthma, for example, or other autoimmune disorders in rural regions or in the developing world. Now, these pediatric autoimmune neuropsychiatric disorders associated with strep infection or PANDAS results from usually acute infection with the group A beta hemolytic strep. So people can have a strep throat, for example, or pharyngitis. In the old days, when people didn't take antibiotics, sometimes those strep infections would progress and that individuals would develop antibodies against strep, and then those antibodies would result in things like arthritis, for example, or myocarditis or rheumatic fever, for example, or they could be associated with Sydenham's chorea movement-related problems or obsessive-compulsive disorder, with the idea being that antibodies that normally would be directed against the bacteria may be cross-reacting with different structures in the brain, resulting in some of these different kinds of symptoms, or cross-reacting with structures in the heart or in the kidneys. In addition to the tic-related symptoms or the movement-related problems or the obsessive-compulsive problems that we see in patients with PANDAS or related conditions, these patients often present with a range of neuropsychiatric symptoms. So they can present with sort of hyperactivity or more restlessness or disruptive behaviors. These patients can have more separation anxiety symptoms or more mood changes or irritability, sleep issues, bedwetting, problems with handwriting, joint pain or inflammation. And so the idea here was molecular mimicry. Proteins on the cell wall of strep bacteria seem to be similar to proteins in regions of the brain like the basal ganglia or the caudate. And so these anti-brain antibodies that bind to the caudate would result in OCD symptoms or motor-related manifestations. Or they could react with the heart or the kidneys, for example. And nowadays, we're doing a study where we're routinely looking at these pro- and anti-inflammatory cytokines and different anti-brain antibodies. So antibodies against NMDA receptors, for example, in the brain, or antibodies that react with basal ganglia, for example. And so you can measure these things in blood work in patients who have a sudden onset of neuropsychiatric symptoms associated with infection. Now, autism is a developmental disorder that has an early age of onset with two core symptom domains. We've always thought about this as being a strong genetic transmission, and that there's a strong concordance in identical twins, for example, compared to non-identical twins. But the recent increases in the rate of presentation of people with ASD, now 1 in 68, have also suggested that there can be gene by environment interactions. So in DSM-5 for autism spectrum, we have two main domains, this social communication domain and the repetitive behavior domain. And you have to have marked impairment in both of those domains to have an autism spectrum disorder. We've progressed from our understanding that there may be one single gene that causes autism to the idea that there may be 100 different genes with small effects. As individuals age, so as fathers age, for example, and sperm sort of replicate over and over, and DNA gets copied over and over, there are more chances for having small deletions or duplications of chromosomal material. And there are a range of different genetic disorders where patients can also present with symptoms that are often identical to patients who have typical autism spectrum disorder. These are the syndromal forms of autism. And now we think of epigenetics, so things in the environment that may shape the expression of genes that may play a role in turning these genes on or turning these genes off that can be associated with behavior in animals or in humans. One cluster or network of genes that seem to play a very important role in autism spectrum disorder is this whole network of genes and proteins related to glial function and immune activation. So genes associated with immune response, wound healing, regulation of cell proliferation, and the inflammatory response. And in fact, in addition to these genes that play a role in the development of synapses, it's these glial and immune genes that are among the most important gene networks in autism. We also know that brain circuits play a particularly important role in shaping behaviors. We know that frontostriatal circuits, or activity of the caudate or the basal ganglia, play an important role in shaping the repetitive behavior domain, both in autism and in a range of other disorders that are classified as obsessive compulsive related or spectrum disorders, and that the bigger the volume of the caudate, the more severe these repetitive behaviors across a number of different conditions. This is our textbook of autism spectrum disorders published by American Psychiatric Publishing. And we talk about some of the experimental therapeutic approaches in autism, and I'll touch on a couple of these today. Some role of oxytocin in both repetitive behaviors and social communication, as well as the role that oxytocin can play in inflammation or wound healing. Another area that's of great importance is the cell cycle. It turns out that individuals with autism frequently have a larger brain volume or bigger brain circumference at critical stages in brain development. So let's say age 12 months to 18 months. And that this particular pathway may be turned on, the cell cycle pathway, that can be associated with things like both macrocephaly, bigger brains, or more dendritic sprouting, and that could also play a role in perseverative behaviors and social communication problems. Now I'm going to talk a little bit about the role of different treatment approaches that may modulate inflammation as a potential new experimental therapeutic approach in autism spectrum disorders. So oxytocin is a peptide that's made in the brain in the hypothalamus, and the neurons reach to the posterior pituitary, where this peptide can be released into the peripheral bloodstream. And release of oxytocin into the periphery can cause milk letdown or lactation, and can play a role in uterine contractions or labor and delivery. But there are a lot of these oxytocin receptors in key regions of the limbic system and in the social brain circuitry. And when oxytocin is released in the central nervous system, it can play an important role in this social communication domain. So things like recognition, social memory, the development of trust. And this oxytocin system also plays an important role in modulating the cell cycle pathway and plays an important role in having anti-inflammatory or wound healing effects. So for example, oxytocin plays a role in social recognition. When animals recognize other animals, they release oxytocin, and that's involved in things like social memory, being able to recognize others. Oxytocin plays a strong role in social affiliation, and so that animals in the wild that are highly social tend to have lots of these oxytocin and vasopressin receptors in brain regions like the limbic system. Oxytocin is also involved in affiliation or the development of trust. And so sniffing oxytocin, for example, may be involved in developing trust or social affiliation with other individuals. And oxytocin also plays a role in modulating social threat signaling. So individuals that are looking at fearful faces often have a threat response because they light up the fear sensor, the amygdala. And when they sniff oxytocin, this seems to dampen down this amygdala threat response. And we have some studies where we've infused oxytocin in adults with high-functioning autism spectrum disorder and found that administering oxytocin, whether intravenously or intranasally, may improve some of the repetitive behaviors that we see in patients with autism, in particular these lower-order self-stimulatory type perseverative behaviors that are characteristic of autism. We've also shown that when we administer oxytocin to young adults who are high-functioning autism spectrum disorder individuals, that those individuals may be better able to lay down certain kinds of social memories, and those social memories can persist over time. Individuals can develop more skills in terms of being able to recognize emotions in spoken language, for example. Now, one important approach now is that we're starting to look at syndromal forms of autism. So rarer forms of autism where we know what the genes are that are involved and we know what the molecular mechanisms are that are involved. And if we can pick individuals where there's a particular gene or a molecular mechanism, that's known, then we can, in a more personalized fashion, match a particular treatment for a particular subgroup of individuals who may be most likely to respond to that treatment. So one of the studies that we're looking at now is in patients who have Prader-Willi syndrome, where they have these small deletions or duplications in regions of the chromosome like 15q11, and where they present with symptoms of compulsive eating and symptoms of autism spectrum disorder, that we might be able to administer things like intranasal oxytocin and find that those are individuals that may have a particularly robust response. And this is particularly true because people with Prader-Willi syndrome have abnormalities in the actual pathology of these oxytocin neurons that go from the hypothalamus to the posterior pituitary and then back to certain reward centers of the brain, so that there seems to be something altered about their oxytocin system. And in those individuals, we might be able to see a bigger treatment versus placebo response, and it may be easier to be able to develop new treatments for specific populations where you know if there's a problem with the oxytocin system and you can identify individuals with this particular problem, then those would be the people that may have a particularly robust treatment effect, and where in a relatively small population you might be able to develop new treatments and show important differences between the active medicine and the placebo medicine in terms of drug development. Now, we've been interested in this observation that some patients who have an autism spectrum disorder, when they have a fever as a result of being sick, there's a clinical improvement in some of the symptoms. So, for example, parents have noted, and maybe 30 to 40% of individuals with autism spectrum disorder, that when they have a high fever, their children, for example, seem to have better eye gaze, or seem to be more affectionate, seem to be more connected, have less irritability or disruptive behaviors. And so we were trying to understand more about what was the possible mechanism for this clinical improvement in certain symptoms of autism in some individuals associated with fever. We thought that, one mechanism was that there was some kind of direct effect of fever on the different molecular or gene mechanisms in the brain. And if there's just a direct effect of fever, then heating individuals to a higher temperature might activate those pathways or activate genes that could be thermolabile or activated at certain temperatures that could be involved in, for example, gene expression or epigenetics. We also thought that it's a possibility that there's an indirect effect of fever, that fever may activate the norepinephrine system. And the locus coeruleus norepinephrine system is a very large brain system that can be particularly sensitive to stress at certain stages during pregnancy, and that mothers that are exposed to severe stress at certain stages of development, certain stages of the pregnancy where the brain is developing, may actually downregulate their noradrenergic or locus coeruleus system, and that medicines that enhance norepinephrine might reverse that. And then finally, that the fever really was just a reflection of some kind of immune inflammatory process, and that if we could develop certain safe or well-tolerated immune inflammatory treatments, that there might be some patients that by dampening down the pro-inflammatory cytokines or decreasing microglia activation, we could get an improvement in certain clinical symptoms. And we used a helminth, a pig whipworm called Trichora suis ovo, TSO, and looked at the effect of this, the helminth or this parasite, which is a parasite found in pigs, but it doesn't cause illness in humans, to see whether or not that might be helpful in dampening down the inflammatory response and whether that would be associated with improvement in clinical symptoms. So the earlier observation was that some patients with fevers seem to have less irritability or social withdrawal. They seem to have less hyperactivity or less stereotypies or inappropriate speech. So what we wanted to do was we wanted to take individuals with autism and first see if we could study them in a hot tub and be able to get them up to 102 degrees. And then we wanted to see whether those autistic individuals who had a history of having a clinical improvement associated with fever, if we took that subgroup and compared how they did when they were heated up to 102 degrees in a hot tub versus when they were in a hot tub at 98 degrees, so sort of a non-fever situation. And then we wanted to see whether there were any biomarkers that might help us to determine better what were the brain mechanisms that might be associated with any kind of clinical changes associated with temperature. So we were able to study 10 children with an autism spectrum disorder who had this history of getting better with a fever. On average, they had an IQ of about 60, and they were about 9 years old. And we found that on the day that we had them in the hot tub, the hydro works at 102 degrees, we were able to elevate their temperature up to almost 102 degrees. Whereas on the day that they were in the hot tub at 98 degrees, and these days were separated by a week or two, that those individuals ended up at a much lower temperature of about 99. So we found that we would be able to get a big difference in terms of the core body temperature in children with autism when we studied them in a hot tub of 102 degrees versus 98 degrees. First, we looked at a global improvement with the Clinician Global Impression Improvement Scale. And we found that there was a statistically significant difference, and that overall patients at the 102 degrees were rated as being significantly improved compared to individuals who were in the hot tub on the 98 degree. So overall, we were getting a global difference in terms of their behavior in these individuals who had a history of improvement with fever. And then we administered a range of other clinical scales. We looked at the social responsiveness scale, total raw scores, and found again that there was an improvement in their total scores on the SRS when they were at a higher temperature of 102 degrees compared to when they were at the 98 degree hot tub day. And we wanted to look at the type of symptoms in particular where we saw some change. And on this restricted and repetitive behavior subscale, we found that individuals with autism at a higher temperature had a significant reduction in the severity of their repetitive behaviors compared to individuals who were in the hot tub at 98 degrees. We also saw some improvement in social domain on this SRS social behavior subscale at the higher temperature versus the lower temperature. So we saw that at a high temperature, patients who had a history of improvement with fever actually had a global improvement in their behavior, in particular in their repetitive behaviors and some of the social responsiveness domain behaviors as well. Now, we weren't exactly sure what the mechanism of this were, and we looked at a range of biomarkers like noradrenergic function by pupillary response or gene expression as well. And now what we're doing is correlating changes on the biomarkers with the clinical improvement, so we understand a little bit more about the mechanisms that are involved in the fever response. Next, it's been discussed that individuals with autism have a significantly increased risk of having a first-degree family member with an autoimmune illness. So there are higher rates of ulcerative colitis or a systemic lupus or myasthenia gravis or rheumatic fever or any autoimmune disease in first-degree family members of individuals with autism. So these immune inflammatory disorders seem to run within the families of patients with autism. Judy Vanderwater, for example, and others have discussed this maternal immune activation response, which can be seen in both animals or in humans. In animals, what can happen is that you can trigger inflammation by either exposing them to the flu virus, for example, or exposing them to the protein coat of the virus, for example, or doing other things that will trigger these pro-inflammatory cytokines like IL-6. And then if you activate inflammation in mothers, when you look at the offspring in animals, those offspring actually have a lot of these perseverative, repetitive behaviors, as well as some social deficits. So that's something that can be seen in the animal model. And we see that when things are measured in the blood, there are antibodies that cross-react to different kinds of brain tissue, like myelin basic protein, for example, or the serotonin receptor, or certain heat shock proteins in the brain. And it turns out that these pro-inflammatory cytokines that can be seen either in the periphery or in the central nervous system may activate this cell cycle pathway. In neurons, activating the cell cycle pathway can be associated with bigger neurons or macrocephaly, bigger brains or bigger head circumference. And in the periphery, activating the cell cycle pathway can be associated with cell proliferation, for example. And it turns out that these cytokines can bind to these PI3K receptors, which can stimulate this PI3K ACT mTOR pathway that can be associated with the growth of, let's say, tubers or lipomas in patients with tuberous sclerosis, or can be associated with the development of tumors, for example, in cancer, for example, and can be associated with neuronal growth in different conditions as well, bigger neurons. Now, it turns out that pumping out a lot of cytokines in the periphery can be associated with increased cytokine signaling in the central nervous system. So things like TGF-beta. And activating complement and cytokine in the central nervous system can be associated with activation of microglia. Activation of microglia in the brain can turn on pathways that are associated with pruning of synapses, for example. So microglia are involved in synaptic pruning, and complement can play a role in modulating microglia activation. Beth Stevens has developed this model that autism and developmental disorders are a disorder of excess synapse elimination, in that an increase in pro-inflammatory cytokines like TGF-beta may be associated with activation of microglia, for example, in the brain, and activation of microglia in the brain can turn on excessive pruning. Turning on excessive pruning at different stages of early brain development can shape brain circuits and brain networks and bias animals or humans towards pathways that can be associated with developmental delay or developmental disorders. If you look at microglia in brain tissue of individuals with autism, there's evidence of microglia activation or neuroinflammation taken in brain samples. And these are individuals studied, let's say, by Carlos Pardo, where they've died as a result of drowning or being hit by a car, and then the brains have been harvested, and then he's looked at the brain tissue, and then he's found evidence for activation of microglia or inflammation or problems in these spatial organization columns in the cortex of individuals with autism. Now, recently, there are these biomarkers that give you an idea of whether or not microglia can be activated in living individuals. And there have been some studies, for example, in Japan with a particular PET ligand that measures microglia activation, where they've compared high-functioning adults with autism spectrum disorder versus matched controls, and they find evidence for this microglia activation. So there are these biomarkers now that can be used that can look at this in living individuals. There are other developmental disorders like Rett syndrome that seem to be associated with activation of microglia. And there are animal models that have been developed of Rett syndrome. Turns out that these mouse models of Rett syndrome, the mice die at a very early age. And people like Jonathan Kipnis have shown that if you irradiate the peripheral immune system with radiation in these mice who have a model of Rett syndrome, so you wipe out their peripheral immune system, and then you just infuse healthy T cells into the periphery. Those healthy T cells or immune cells can cross the blood-brain barrier and get engrafted into the brain, and they can modify or arrest the microglia pathology in mice. And these mice actually go on to live a normal lifespan as opposed to die at a very early age. So one idea is that you can modulate the peripheral immune system, and that could play a role in the central nervous system as well in individuals with developmental disorders. So there's this immune dysfunction hypothesis in autism spectrum, which comes from this hygiene hypothesis. The idea is that in rural areas like Iowa, where there are a lot of favorable gut parasites that are seen in the soil, that are common as a healthy gut bacteria in individuals in rural areas, associated with those favorable gut parasites are low rates of things like asthma or autoimmune disorders or autism. And interestingly, in the CDC estimates suggest that the more urban states have higher incidence or prevalence of autism spectrum disorder versus rural states. Although there are a lot of complicating factors that may influence that. It may be that there's more screening done in urban states for autism spectrum disorder, or that individuals move to urban states to get services. But the idea is that in more rural states, where there's more favorable gut parasites, for example, that this may dampen down chronic immune activation, and decreasing chronic inflammation can be associated with less autoimmune problems and possibly less developmental disorders. As I mentioned, both in the spinal fluid and in the brain of individuals with autism, you see a lot of these pro-inflammatory cytokines and evidence for activation of microglia and astroglia. So administering something like TSO, a Tuberous suis ova, which is a pig whipworm, which doesn't reproduce in the human gut, so you have to give it every 2 weeks, and it doesn't penetrate the intestine, so it doesn't cause medical illness like anemia that other parasites do, that it may be able to trick the human immune system by decreasing pro-inflammatory cytokines, increasing anti-inflammatory cytokines, shifting the T helper to T suppressor ratio. And there have been studies with TSO and other autoimmune illness like inflammatory bowel disease or multiple sclerosis or things like peanut allergies. So, for example, in ulcerative colitis, it seems to have a positive effect of decreasing these manifestations of inflammation. So we did a study in 10 young adults with high-functioning autism spectrum disorder. I'm going to show you the data from the first 5 patients who completed both of the phases of the trial. So the first phase, patients were either started with the helminths or with the placebo for 12 weeks. Then they had another 4 weeks or 1 month where it was washed out. And then they had another 3-month period where if they were started on the active medicine, they were switched to placebo or vice versa. We specifically picked young adults who had high-functioning autism with a normal IQ and good verbal skills. And we also selected for those individuals who had a history of seasonal allergies or other types of allergies, and who have this sort of family history of other autoimmune illness, so like family history of myasthenia gravis, for example, or inflammatory bowel disease or lupus. So these were young adults, high functioning, and So we did some analyses on these, and we looked at a number of measures that we had developed to tap into the repetitive behavior domain that's seen in individuals with autism spectrum disorders. I mentioned that in order to get the diagnosis of autism, you have to have increased narrow restricted interests or rigid routines or repetitive interests, and as well as the social communication domain. And it turns out that this early rigidity is a sort of an early marker of autism. That those individuals who have severe early rigidity and repetitive behaviors are the individuals who are most likely to meet criteria for the full autistic disorder down the road. So it's really one of the two core features that you see in autism. We developed a Montefiore-Einstein rigidity scale that in particular measured whether or not individuals are very rigid, whether they experience discomfort when there are unexpected deviations in their expectation, and whether or not that discomfort associated with deviation and expectation is associated with protest or irritability. And what we saw in these individuals who were treated with the TSO helminths, which decreased the pro-inflammatory cytokines, was that there was an improvement in the rigidity scale scores that it took some time. So we didn't really see any effect over the first 8 weeks, but from weeks 8 to week 12, what we saw was a big decrease in rigidity when they were taking the TSO and no change when they were taking the placebo, and a significant, statistically significant separation on this measure of rigidity. We also have a scale on the repetitive behavior scale score for craving for sameness. And we also saw sort of a delayed response, but then after 10 or 12 weeks, what we were seeing was that individuals who got the helminths that had these anti-inflammatory properties, it decreased the cravings for sameness compared to those individuals who are on placebo. And then also on this SRS repetitive behavior scale score, also after around weeks 8, 10, and 12, that those individuals who are on the active Tricura suis ova helminth compared to those people who are on placebo, where there was a statistically significant reduction in their repetitive behavior. So what we saw was on 3 different measures that looked at things like rigidity, craving for sameness, and repetitive behaviors, that over time, after around 8 weeks or so, what we were seeing in those individuals who were taking the helminth was an improvement in that domain compared to those people on placebo where there was no change. These people didn't have high irritability at baseline, and there were only relatively modest effects on the irritability scale score. We are in the process now of being involved in a larger study looking at children who have autism spectrum disorder who have high levels of irritability at baseline as well as a lot of rigidity, and specifically seeing whether those individuals with higher irritability might have a bigger improvement on the irritability subscale scores. So in this pilot study and in our interim analysis, we were seeing statistically significant and clinically meaningful changes on 3 different measures that looked at rigidity, craving for sameness, and repetitive behaviors. We had only a small to modest effect on irritability, but these patients were high-functioning adults and had very little irritability at baseline. We plan to be involved in a larger study now in children who were selected for having high irritability at baseline. In these individuals, it seemed to be well tolerated. None of them had any kind of serious adverse events, and none of them needed to be discontinued due to safety-related issues. It seemed to be very well tolerated in this population. But clearly, these results are in a small population. This is an interim analysis. We need to replicate it in larger sample sizes. And we also want to look at whether or not if we intervene at an earlier course in the illness, so when children are younger and when they have higher baseline irritability, whether we may be able to see bigger overall changes in the population. We do think that there are important gene-by-environment interactions that occur in developmental disorders like autism spectrum disorder, but may also be playing an important role in a range of different neuropsychiatric disorders. Now, we think that there may be changes in the microbiome with less of these sort of favorable or healthy gut bacteria, and that changes in the microbiome can, in a sense, be changes in environment resulting in these gene-by-environment interactions in those individuals who have certain genetic vulnerability. We think that these changes associated with chronic inflammation can be associated with a number of medical illness in highly developed countries, and chronic inflammation can be associated with obesity and risk for type 2 diabetes, risk for things like autoimmune illness and asthma, and that we might have a whole range of different experimental therapeutics that might play a role in modulating the immune inflammatory response. Each of these different things have a different risk versus benefit ratio. So picking therapeutic agents that have fewer side effects may be very important because we can more rapidly sort of advance those treatment trials into humans, and also where we could study children earlier in the stage of the illness or earlier in the developmental process. So I do think that developing sort of novel immune inflammatory modulatory agents that have a good safety profile that can be rapidly moved down to younger individuals earlier in the stage of the developmental process may be very important. And that ultimately some of these agents may work by influencing cytokines both in the periphery as well as in the central nervous system, and that affecting cytokines in the periphery and in the CNS may have a role in microglia activation. We think that microglia activation may play an important role in different neuropsychiatric disorders and may play a critical role in developmental disorders, and that microglia activation may play a role in pruning, for example, and the development of basic brain circuitry and synaptic connections that may be involved in many different developmental processes. And ultimately, our goal is to develop more personalized treatments for autism spectrum disorder. And partly that means stratifying the population. One of the ways to stratify the population is to select individuals who have particular symptoms. So those individuals that have very severe rigidity or craving for sameness, or those individuals who have very severe irritability at baseline, might be one way to stratify the population. Another way to stratify populations, for example, might be to select individuals who have considerable allergies, seasonal allergies, or allergies, for example, to foods or to other things in the environment, or to stratify for populations where there is strong first-degree family members with lots of autoimmune illness that may suggest genetic vulnerability for certain immune inflammatory pathways. The other way to do this is to select more homogeneous populations based on known genes. So, for example, to select syndromal forms of autism, as an example, patients with Prader-Willi syndrome, for example, who present with symptoms of an autism spectrum disorder, or patients, for example, with tuberous sclerosis who have symptoms of autism spectrum disorder, or patients with Rett syndrome that also have symptoms of an autism spectrum disorder. So to focus on rarer homogeneous syndromal forms of autism, to develop new treatments or experimental therapeutics that may modulate the immune inflammatory system in those more homogeneous genetic populations. The other thing that's extremely important is to develop good early efficacy biomarkers. So biomarkers are measures in between the underlying genes and the observable behavior. So biomarkers can be things like eye tracking. So eye tracking may be a good biomarker for the social communication domain. Or, for example, certain EEG measures may be a biomarker. Measuring cytokines, either in the periphery or in the spinal fluid, may be a biomarker, for example. And we want to use these biomarkers because we want to demonstrate that any of our new experimental therapeutics are actually engaging relevant targets, like the correct brain circuitry, for example, or EEG patterns that are easily measurable, or eye tracking, for example, or that may be affecting biomarkers like microglia activation that we can measure, for example, using PET ligands in humans. And if we can develop more personalized treatments for rare or homogeneous populations or syndromal forms of autism, and we can show engagement with these early efficacy biomarkers, and we can stratify the population for those populations where there may be the best fit between the underlying molecular mechanism and the experimental therapeutic target, then we can develop new treatments in relatively small populations. And if we show a big drug versus placebo signal, then in these rarer or stratified populations, then we can then see whether or not these treatments may be applicable for the larger population with autism spectrum disorder as well. So I've talked a little about the role of inflammation in autism and related conditions, a little bit into the idea of experimental therapeutics to try to target inflammation in patients with autism. And I've talked about a couple of simple strategies that can be very helpful in terms of drug development that may have a big impact by modulating this system, and that may be applicable to a broader population with autism spectrum disorders as well. I wanted to thank ARI for inviting me here, and I'd be glad if there's some time to take some questions as well. Thank you.
Hi, Dr. Hollander. Yes, we have a lot of questions, so I will try to keep them. I'll try to cover several questions in the ones that I give to you. I think one of the first questions I want to ask that a lot of people submitted was they want to know more about how you would test for inflammation. Are there blood tests? Are there stool tests? What kind of testing might they do to learn more about where their child or loved one is at?
Well, so one is you can see whether or not they have any other manifestations of inflammation. So allergies, for example, asthma is an example, other evidence for activation of the immune inflammatory system, other autoimmune disorders, for example. There can, as I mentioned, there can be simple measures in the blood that clinicians can do. So people can look at things like C-reactive protein or ESR, erythrocyte sedimentation rate, and you can measure some pro-inflammatory or anti-inflammatory cytokines in the periphery. And those are sort of readily available and can be done in commercial labs. We and others are also starting to look at antibodies in the blood that may be anti-brain antibodies, because if you can measure antibodies that can be binding to different structures in the brain, that may be important and may tell you whether or not there could be some inflammation and guide you in terms of selecting some treatments. As I mentioned, there are these more exploratory type of biomarkers that can give you a measure of inflammation. And so one of these biomarkers that's been studied in Japan is this PET ligand for astroglia activation, so that in patients with autism spectrum disorder compared to individuals who are healthy controls, there was some evidence for sort of activation of microglia with this particular PET ligand, for example. So I think, one would be in taking a good history, looking for other sort of manifestations of inflammation or other medical illnesses that might be associated with inflammation. Two is by doing simple, common, laboratory measures like C-reactive protein, because that's been shown to be helpful in other mood disorders or even in schizophrenia, in suggesting that the subgroup of individuals with high C-reactive protein seem to have some positive response with immune inflammatory modulating treatments, for example. Looking at things like pro-inflammatory, anti-inflammatory cytokines, which can be measured in the periphery, which they can also be measured in spinal fluid or in the brain. And then a good sort of discussion of family history and whether or not there are these high rates of other immune inflammatory illness or other sort of markers of inflammation that are seen in other family members.
Okay, the next question is about using novel therapeutics now. So people are, had a lot of questions about acquiring oxytocin or TSO. Are those therapies available now? Are people using them?
Well, first I would say that neither oxytocin nor TSO are approved by the Food and Drug Administration for the treatment of autism. And in fact, there are no treatments currently that are approved by the FDA for the treatment of core symptoms of autism. There are 2 treatments that have been approved by the FDA for the treatment of a small subgroup of individuals who have an autism spectrum disorder who have severe disruptive behaviors, aggression or self-injury, and those are the atypicals, risperidone and aripiprazole. So in a sense, we're really talking about mostly off-label treatments. There have been a number of clinical studies using intranasal oxytocin to treat individuals with autism, but other conditions as well, including schizophrenia and social anxiety disorder, showing that in some clinical measures there's an improvement in social communication domain, as measured by a number of different clinical measures, and that the intranasal oxytocin seemed to be well tolerated in these populations. However, I would say that there haven't been any large-scale studies in autism where intranasal oxytocin has been developed and shown to be safe and effective in large-scale pivotal trials that have been presented to the FDA for licensing. And so that kind of large-scale data doesn't exist yet. With regards to TSO, there have been placebo-controlled trials of TSO in different immune inflammatory conditions, showing that it seems to be pretty well tolerated in these different populations. And then we have this relatively small-scale study in a group of patients who had high-functioning autism and good verbal skills, who were selected because they had a history of allergies and family history of immune inflammatory illness, where we saw some improvement in 3 different measures of this rigidity domain, and that there are some other studies that are about to be launched in children with autism. But again, there haven't been large-scale pivotal trials done yet in patients with autism, and there's no FDA indication for the use of TSO for autism or any other illness in the United States either. So I just wanted to provide that background as well. So there are individuals who have used both of these treatments in off-label treatments with some success.
So one more question along those lines. One parent is asking about the use of minocycline and pycnogenol as potential treatments. Have you seen any research on that?
Well, so minocycline is an antibiotic that's often used to treat acne, and there have been some trials with minocycline now in children with autism spectrum disorders and also individuals with OCD and other conditions. In those clinical trials, there wasn't overall a separation, so that the active medicine didn't statistically separate from the placebo. There are, minocycline has been shown to have some immune inflammatory regulating properties. And in fact, it's interesting, one impact that antibiotics may have is, they kill off bacteria like strep. But, antibiotics also can have some immune inflammatory modulating properties that may be independent of their antibiotic properties. I would say that with regards to the data, the one placebo-controlled trial that was done in childhood autism didn't show any statistically significant separation from placebo.
The next question is about study enrollment. A lot of parents are interested they want to know more about getting active in studies that you're running, or if you know any other studies that they might investigate.
Well, so one way to look at that is to go onto clinicaltrials.gov, and they often have a list of different clinical trials if you put in the particular disorder, and that can provide one way to look at clinical trials. I mentioned that we're in the process of being involved in a multi-center study to look at TSO in children who have autism spectrum disorder, who have high levels of irritability at baseline. And so at Montefiore and Albert Einstein, we will be enrolling patients for that study, as well as we have other trials looking at vasopressin 1A antagonists, for example, in young adults who have high-functioning autism, and we're looking at lurasidone for children who have autism spectrum disorder with a lot of disruptive behaviors. Coronado Bioscience is launching this 3-site study in children with autism with TSO, comparing it to placebo. So that's a clinical trial that's going on as well.
Okay, another question. They are asking, I'm not quite sure what they're asking, so you may have more insight about this. They're asking, is inflammation causative or is it related?
That's a good question. Because you see inflammation in some individuals with autism, the question is, well, is this something that is associated with but doesn't play a particular role in the development of the disorder? Another question is whether or not sort of immune inflammatory mechanisms, they may play a role in the early stage in brain development, but are they still playing an ongoing role as individuals get older? And large-scale epidemiology studies often show like an association. So I talked about those studies, for example, that show that if you have recurrent infections and you have autoimmune illness, your risk of developing a depressive disorder was high, and that there was an interactive effect. But again, that's showing an association as opposed to causation. The animal studies of this maternal immune activation, where in animals you trigger inflammation in the mothers during the pregnancy, and you show that the offspring have problems with a lot of rigidity and repetitive behaviors and some social difficulties. And then you administer to the mothers antibodies to the pro-inflammatory cytokines like IL-6, and then show that the offspring don't have the behavioral problems. I mean, that would suggest a causation role, at least in this animal model of maternal immune activation. That Judy Vanderwater has studied autoimmune sort of antibodies in mothers who had offspring with an autism spectrum disorder, and I would say that that would be a association, whereas in an animal model, you're more likely to be able to demonstrate causation because you can you can modify a factor, show that that causes illness in the offspring, and then counteract that factor and show that the offspring don't have the illness. Same in the animal models, for example, with Rett syndrome. In a particular gene that's associated with sort of microglia activation, you can wipe out the immune system in the periphery and infuse healthy T cells, and then all of a sudden these mice that are dying at a young age live to a much longer age. It suggests that modifying the peripheral immune system, plays an important role in the disease progression and the lifespan in that animal model, for example. So sometimes you turn to animal models to help answer that question of causation, and that epidemiology studies probably give you more information about association. Treatment studies can suggest that if you intervene with a particular treatment, that you can improve certain clinical symptoms of the illness. What you'd also want to show is that that clinical treatment is engaging the right target, and that by affecting that target, that's influencing the mechanism of the underlying illness. And so, one of the ideas in experimental therapeutics is not only to show that you can give a treatment and improve symptoms, but that you're also having an impact on the system that's involved in mediating those symptoms. And that would be more reflective of causation as well.